Welcome back, everybody. We're going to jump right into Chapter 4. Let's talk about blood lipoproteins. So to this point, we've kind of talked about cholesterol, a little bit of the breakdown of the fats, kind of how we're using those bile salts and everything to be able to absorb those fats. And right now we're going to talk about the blood lipoproteins, which are really going to be involved in the transportation of these different fats throughout the bloodstream, throughout the body. So at the end of this lecture, we will be able to recognize the importance of the lipoproteins, classify various types of lipoproteins, and know their general composition, explain the role of major apoproteins specifically present in various lipoproteins, indicate the site of biosynthesis and sources of blood lipoproteins, demonstrate the mechanisms of transport lipids demonstrate the mechanisms of transport lipids carried via various blood lipoproteins, describe the metabolism of blood lipoproteins, and explain the major function of blood lipoproteins. Describe the metabolic fate of the blood lipoproteins. Okay. So one major source of lipids is our diet. Lipids are also synthesized by the liver and adipose tissues. Various types of lipids must be transported to target tissues and organs for utilization to produce energy or to make certain essential molecules. Lipids are also stored in tissues for future utilization. Now, how lipids are transported through the circulation and organ systems. Most lipids circulating through the body are captured and bound by lipoproteins and then delivered to the tissues where they are metabolized. Some simple free or unesterified fatty acids are circulated via binding to plasma albumin. Plasma albumin is one of the most important proteins that is in the blood for maintaining our blood volume and for maintaining kind of the osmotic pressure of our blood. You'll learn more about that in physiology. Binding to plasma albumin, a non-specific but important and the most abundant protein in blood plasma. Other lipids, such as triacylglycerols, TGs, remember triacylglycerols are the same thing as triglycerides. Phospholipids, cholesterol esters, remember this is the storage form of cholesterol, are transported by lipoproteins. However, there is one problem. Most lipids, or a portion of lipid molecules, have a hydrophobic, nonpolar nature. Thus, lipids are insoluble in the aqueous, or the water environment in the circulation of the blood. This problem is solved by combining the nonpolar lipids, such as the triacylglycerols or the cholesterol esters, with amphipathic lipids. Amphipathic lipids, meaning that they have polarity, such as the phospholipids or cholesterol, and adding some other proteins known as apoproteins, and this having more polarity or electrical charges. This procedure makes the lipids more soluble in the aqueous medium of blood. So we're going to take those triacylglycerols and the cholesterol esters, and we're going to combine them together with the amphipathic. Let me real quick. Amphipathic. So amphipathic is defined as having both the hydrophilic and hydrophobic parts. So stuff like the phospholipids and the cholesterol have both hydrophilic and hydrophobic parts. So we combine the completely hydrophobic with the partially hydrophilic, additionally to some of the proteins, which then have the different polarity electrical charges to allow them to then be aqueous, be able to travel in the blood, which is primarily water. So the biomedical importance of lipoproteins. As carrier, transporter, or mobilizer of lipids such as cholesterol, cholesterol esters, triacylglycerols, and other lipids, those phospholipids, sphingolipids, ceramide, etc. To store energy in the form of lipids in the liver, intestine, and adipose tissues, etc. To utilize lipids by oxidation within muscle tissues, the skeletal, and the heart to release energy. It's what we talked about, beta oxidation, breaking those down to create the components, the acetyl-CoA, which can then go into the Krebs cycle. That's a question for you. You can think about that. So in order to burn fat, should the body be in an aerobic or an anaerobic state? So if we think about whenever we're burning fat, if we're releasing acetyl-CoA, and then acetyl-CoA is going directly into the Krebs cycle. The Krebs cycle and the electron transport chain all require oxygen, so it's going to be oxygen requiring in order to burn fat. That's actually why technically when you work out, there's sort of a sweet spot heart rate, which is recognized as the rate at which you are primarily fat burning. Which, depending on what your exercise goals are, maybe what exercise goals your patients have, you can discuss with them. But that's why we typically, when bodybuilders and people are on a cut and they're doing cardio, you'll see them low speed, high incline, because they're aiming for a specific target heart rate. And that specific target heart rate and that specific level of activity allows the body to be running not just on anaerobic glycolysis, but having the aerobic breakdown of fats in order to cut, or specifically to target fat loss. So it's kind of cool that when you get, that's what my degree kind of focus on, is exercise physiology, exercise science, and exercise physiology, really getting into the nitty-gritty of that and kind of the calorie burning, or what kind of calories you're specifically burning. So that's really, really interesting. A really interesting topic that I enjoy looking into. Okay, continuing. So abnormalities in lipoprotein synthesis, storage, receptor function lead to various diseases. Hypertriacylglycerolemia. So if we break this down, hyper versus hyper meaning too much. Triacylglycerol, we're looking at triglycerides. Heme has to do with the blood, so this is high triglycerides in the blood. Hypercholesterolemia, same thing. Hyper meaning high, cholesterol being cholesterol, emia in the blood, high levels of cholesterol in the blood. Atherosclerosis, we talked about what that is. If you don't remember, go back right now and look at it. As well as obesity. Obesity is going to be defined as more so than just being like a specific weight. It really is more of a fat percentage. What percentage of your body mass is made up of adipose tissue versus solid muscle. So what are lipoproteins? So lipoproteins are lipid plus the apoprotein. Remember earlier we saw the definition of the apoprotein. The apoprotein is going to just be other additional like regular proteins. We call them apoproteins that are going to be added to or be joined with the lipids and the amphipathic lipids as well, in order to make them able to travel in the aqueous solution of the blood. So apoprotein is the protein moiety of the lipoprotein in its lipid-free form. So it's the protein that's all been folded up and everything of that. What the apoprotein is, the protein part of the lipoprotein in its lipid-free form. So we have the lipids are insoluble, the proteins are water-soluble. You get lipoproteins, which are now water-soluble. So you have a lipid core with the water-soluble exterior, and they can move and transfer and enter different cells and go through the blood. So besides having specific functions, apoproteins also increase water solubility of the lipoproteins. Consists of a core. This is important to remember that the core of hydrophobic lipids surrounded by a shell of polar lipids and apoproteins. The principal apoproteins in various lipoprotein particles are ApoA1 to 4, ApoB48, ApoB100, ApoC1, ApoC2, ApoC3, ApoD, and ApoE. The composition of these apoproteins may vary from one lipoprotein type to another. From what I recall, this isn't super important to remember each of these individual names. Just being able to kind of understand what an apoprotein is, and that they're made up of different protein components. So lipoproteins are composed of various combinations of polar lipids with insoluble lipids, and thus these lipids can be transported within a specific tissue to perform their specific functions. For example, muscle tissue oxidizes lipids in the form of free fatty acids and produces energy. Adipose, liver, and mammary tissue store lipids for future use as sources of energy. For example, the lactating mammary tissue also uses stored lipid for milk production. You'll learn a lot more about that in your pediatrics class. That class, you talk about kind of like the different stages of milk production and the different quantities and levels of carbohydrates and proteins and fats in milk that allow for the proper growth and development of the babies at different periods of time. Okay, so two main roles of lipoproteins is solubilize or make soluble the highly hydrophobic lipids. And they also contain signals that control movement of particular lipids into and out of specific target cells and tissues. They are the major transporters of various lipids. So the major transporters of various lipids into the tissues, we have cholesterol, triacylglycerols, and other lipids are carried in body fluids by the lipoproteins. Lipoproteins are classified according to their increasing density. So we'll start, we have the chylomicron, and then we have chylomicron remnants. So remember we talked about the chylomicron and chylomicron remnants. Chylomicrons are found in the small intestine when they grab onto the triacylglycerols coming from the small intestine after they've been digested, and they'll go through and then they'll get broken down and they'll become chylomicron remnants. And then you have very low density lipoprotein, which means this has a lot of fat in it, as well as protein. Then it goes to intermediate density lipoprotein, low density lipoprotein, and high density lipoprotein. So kind of here on, we're looking more at, this is more in the liver, and then our LDLs and our HDLs more in the blood. They are all throughout the different structures of the body, but we tend to identify them with the chylomicrons. We tend to identify more with the small intestine, the VLDLs more with the liver, and then the HDLs, LDLs with the blood. So the major composition of the lipoproteins, we have triacylglycerols, cholesterol, cholesterol esters, phospholipids, and proteins. So the detailed composition of lipoproteins are described in the following two tables. So this first table is the density of the lipoproteins and their total protein and lipid content. So this is just kind of showing you a little bit more exactly kind of what we were talking about. You don't worry, you don't really need to memorize any of these numbers, just understanding the pattern and the order of these. So the chylomicron being in the intestine, VLDLs in the liver and the intestine, the IDLs, they come from the VLDL and the chylomicrons, LDLs come from the VLDLs, and HDLs are from kind of all over, but we'll see them a lot in the blood. The reason why you have the question mark here, just because like there is that correlation between all of them, but not sure quite exactly like how correlated, depending on who you ask really. will depend on the answer that you get with like how correlated everything is. And here again, the percent total of lipids, you can see all the way through. Of the different fats that we talked about. I don't know why they didn't include proteins in here, percent of protein. That's fine. So the structure of the blood lipoprotein, here we have the blood lipoprotein. We can have cholesterol in there. The apoprotein, B100 is one of the examples. The core of those non-polar lipids, the triacylglycerol is in the middle, a monolayer of mainly amphipathic lipids, cholesterol ester, cholesterol in there, all of those different types of fats all sort of combined with the apoprotein and having more of that amphipathic lipid layer on top. Lipoprotein and lipid transport, we're talking about chylomicrons. These are the largest of the lipoproteins with the lowest density, meaning that they have a much higher ratio of fat compared with protein. They're the intestinal epithelial cells synthesize the intestinal epithelial cells synthesize chylomicrons. They have a very high triglyceride content, which is 88% of total lipid. They contain the highest levels of triglycerides among all the lipoproteins. They're a major carrier of dietary fats to blood via the lymph circulation. Remember this is really important when understanding the absorption of fats. It's via the lymph circulation. And they also have the lowest phospholipid content of 8% of the total structure. Major apoproteins of the chylomicrons are ApoB48, ApoC2, and ApoE. Triglycerides, cholesterol esters obtained from the diet are transported from the intestine to adipose tissue and the liver by the chylomicrons. Triglyceride and chylomicrons are hydrolyzed within a few minutes by lipase. So lipase is going to be the enzyme that's going to break apart lipids located in the capillaries of adipose and other peripheral tissues, producing chylomicron remnants. The chylomicron remnants are taken up by the liver. Okay, so that's how we go from chylomicron to the chylomicron remnants, and then the chylomicron remnants are taken up by the liver to be repurposed and kind of either break them down or to repurpose them and reuse them. Kind of re-put everything together because the body's going to be very efficient in what it is trying to do. Functions of the apoproteins of the chylomicron. So ApoB48 is responsible for assembly and secretion of the triglycerides and other lipids in the chylomicron. ApoB48 is recognized by the LDL receptors required for uptake of chylomicron remnants at the liver cell membrane. Ooh, apologize. ApoC2 acts as an activator, released from lipoprotein, and activates lipoprotein lipase, also known as LPL. It's an enzyme which is localized at the membrane of capillary endothelial cells. ApoC2 is localized in the outer surface of the chylomicrons. Enzymatic action of LPL activated by ApoC2 produces the free fatty acids and glycerol from triglycerides, as well as the chylomicron remnants, the triglyceride-depleted chylomicrons. About 90% of triglycerides are digested and lost. ApoC2 is lost as it is digested by protein breakdown by other enzymes. Free fatty acids are released in the bloodstream and transported in the target tissues. Within the muscle tissues, the fatty acids are oxidized and produce energy. Within the other ones, they're stored. Use of energy, we saw that already. So now ApoE serves as a ligand. Remember what ligand is. Ligand is the chemical, this chemical type structure or whatever it is. It's going to go and bind to the receptor. Remember, this will be really, really important for Physiology 1, understanding what a ligand is and the different types of ligands. So ApoE serves as the ligand bound to chylomicron and chylomicron remnants and detects LDL receptors, which are mainly localized in the liver cell membrane. So those LDL receptors mainly localized in the liver cell membrane. Chylomicron remnants are taken up by the LDL receptors when ApoE detects these receptors and form a receptor-ligand complex. These receptor-ligand complexes are endocytosed, meaning they're brought into the cell, by receptor-mediated endocytosis, trapped into endocytic vesicles, and then they fuse with lysosomes. So we think back a little bit to kind of our cells and tissues. Let me pull up a good picture of what a vesicle. A vesicle is kind of like a sac. This is what it's going to look like. And it has a little bit of a membrane so it can kind of carry stuff in with it. So it's going to be kind of containing it within. And then the lysosomes are digestive proteins. This is the digestive system of the cell, serving both to degrade material taken up from outside the cell and to digest obsolete components of the cell itself. It's going to be there for breakdown. Kind of digest. It's like the stomach of the cell, digesting and breaking stuff down. So hydrolytic enzymes of this new fused vesicle containing lysosomal enzymes digests the receptor ligand complex and produces free amino acids, which are recycled. Next, we have our very low-density lipoprotein, our VLDLs. Excessive dietary carbohydrates and fats are converted to triglycerides in the liver. The VLDLs are also very rich in triglycerides. They're the third in the scale of density, next to the chylomicron. It also contains cholesterol, about 7%, and cholesterol esters. The major apoproteins of VLDLs are ApoB100, which deals with the assembly and secretion and detection of LDL receptors, and the ApoC1, which is an unknown or inhibit hepatic uptake of chylomicron or VLDL remnants. Here's our structure of our VLDL. Got all different components. ApoC2 activates lipoprotein lipase. ApoC3 inhibits LPL, and the ApoE ligand. ApoE is the ligand for binding to the LDL receptors to bring it back into the cells. Endogenously biosynthesized triglycerides, in contrast with those obtained from dietary sources, are transported by VLDL. So the chylomicrons are going to be transporting the ones from the diet, and the VLDLs are going to be transporting the ones that the body is producing. Fate of VLDL triglycerides, so the enzymatic activity of LPL, which is activated by ApoC2 and attached to the VLDL. So the enzymatic activities of LPL of the capillary endothelial cells releases triglycerides from VLDL, producing VLDL remnants, or IDL. The IDLs just have less, that's the intermediate density lipoproteins. They have less triglyceride content than VLDLs, free fatty acids, and glycerols. Free fatty acids are transported to respective tissues for oxidation, such as the muscle tissues, and for storage of the adipose mammary tissues, just like we talked about earlier. So here it is, the fatty acids, the VLDLs, giving off the fatty acids and getting uptaken into the muscles and other places. And then we end up with IDLs, or we keep going down, breaking those down, taking more triglycerides off, and we get LDLs, and so on. The intermediate density lipoproteins, the IDLs, are rich in cholesterol esters, so 50% of these IDLs are taken up by the liver by receptor-mediated endocytosis and further digested by lysosomal hydrolytic enzymes and releases free amino acids, cholesterols, fatty acids, glycerols, inorganic phosphates, etc. Another 50% of these IDLs are converted to LDLs by loss of more triglycerides. Low-density lipoproteins, LDLs are our major carrier of cholesterol at 10% and cholesterol esters. The major role of the LDL is to transport cholesterol to peripheral tissues, regulate de novo cholesterol synthesis in these sites. So we talked about how we build cholesterol. De novo just meaning new, so it regulates new cholesterol synthesis in those different sites. Remember, if we have that negative feedback loop of cholesterol. So if we have cholesterol, then it's going to inhibit the HMG-CoA from continuing to produce the HMG-CoA synthase to continue to produce cholesterol. Okay, check. We're getting close to the bottom here. Okay, so LDL receptor-mediated endocytosis. LDL, IDL, chylomicron remnants are detected by LDL receptors localized in the hepatocyte cell membrane. Apoprotein E. So when I said earlier about not necessarily needing the names as much, the names that are consistently brought up, kind of like the apoprotein E here, those ones, if you're going to learn any of the specific names, these are the ones that I would focus on, the ones that get brought up constantly, and just what their function is. So apoprotein E present in these lipoproteins detect this LDL receptor on the hepatocytes. Following LDL and LDL receptor binding interaction, these lipoprotein remnants are endocytosed in the coated pits present in the membrane, which eventually are invaginated to form coated vesicles or endosomes and are engulfed by the lysosomes. So what this is saying is that the chylomicron remnants are going to come up to, let's say this is the membrane right here. Let's say this is the membrane. This is the membrane, the outer membrane of the cell. This chylomicron remnant is going to come, it's going to bind a receptor here, and it's kind of going to curl in like this and form a vesicle with everything inside of it. And then the rest of the membrane is it's going to almost like pinch off a bubble of the membrane to let the membrane go through. So let me actually see if I can find a good picture of this. Like this, where it's binding here, it's coming in, it's kind of creating this bubble right there, and now this is our vesicle that is now inside, and our membrane is still complete, and then we have a vesicle, and that vesicle is able to go in, and then it goes over to the lysosomes. So it is a coated vesicle, also known as an endosome, and those are engulfed by the lysosomes, so those digestive enzymes. Lysosomal enzymes hydrolyze and metabolize these chemical components further. Approximately within 24 hours, most of the LDL are removed from the circulation in this manner. LDL receptors are recycled back to the hepatic cell surface, and there's a new protein of interest called proprotein convertase subtilisin kexin type 9, or PCSK9. PCSK9 was first described to have a major role in the metabolism of LDL when a mutation in this gene was observed to cause a rare form of hypercholesterolemia. Follow-up research further demonstrated that both normal and mutant form of PCSK9 promote degradation of hepatic LDL receptors and also degrade these LDL receptors. So this is something that we'd be referring to, this mutation of this gene, is why you can say that in certain groups of people, hypercholesterolemia runs in their family. They might have some sort of mutation to this gene that gets passed on. Okay. Thus, although PCSK9 is playing an important metabolic regulatory role in hepatic clearance of circulatory form of LDL, these receptors are never recycled back to the hepatocyte surface. The point to note here is that PCSK9 is removing the circulatory LDL and as well helping to the destruction of the LDL receptor. Hence, we are losing available LDL receptors eventually. Now, if an inhibitor can be used to block the PCSK9, then it will be then it will increase the availability of LDL receptors in the hepatic cell membrane and could be more effective in removing circulatory LDLs without further destruction of LDL receptors. The clinical correlates, we already talked about atherosclerosis. Pause, go ahead and reread that again. High density, now our high density lipoproteins are HDLs. So we're going to talk about the synthesis of HDLs. First, nascent HDLs are produced by the liver and intestine as a small molecule. Nascent HDLs are discoid and contain less phospholipids, ApoA, free cholesterols. Nascent HDL does not contain the ApoC or ApoE. Mature HDLs have more globular shapes and receive ApoC, ApoE, ApoD, phospholipids, LCAT, and cholesterol. The HDLs are protein-rich particles and often exchanges protein or lipids with other lipoproteins. The HDLs contain 5 to 10% cholesterol, around 30% cholesterol esters, 13 to 16% triglycerides, and less than 2% free fatty acids, ApoA1, ApoC1, and ApoC2. Mature HDL also serves as the repository of ApoC2 and ApoE for nascent chylomicron and nascent VLDL, and in turn converts the mature VLDL and mature LDL respectively. When we're talking about nascent, we're talking about the newly formed nascent. It's just coming into existence and beginning to display signs of future potential. That is what nascent means as we're reading through that. It uses that word a lot in that little chunk. Okay, so HDLs also receive ApoE. Actually, I think I skipped a line. HDLs pick up cholesterol released into the plasma from dying cells and from membranes undergoing turnover. HDLs also receive ApoE and ApoC2 following VLDL triglyceride digestion. An acyltransferase known as lecithin cholesterol acyltransferase, or LCAT, in HDL esterifies these cholesterols. The LCAT is activated by the ApoA1. So esterified cholesterols are then rapidly shuttled to VLDL or LDL by a transfer protein known as the cholesterol ester transfer protein, or CETP. This is just showing a picture of that, the cholesterol with LCAT going to the cholesterol ester. HDLs either directly transport cholesterols and cholesterol esters to the liver or transfer cholesterol esters to other lipoproteins via the cholesterol ester transfer protein, CETP, also known as ApoD. The major roles of HDL is the delivery of cholesterol to tissues, pick up extra cholesterol from extrahepatic tissues. This is a big one. This is why HDL is considered the good type of lipoprotein, because it's going to be cleaning up the extra cholesterol, so it's not just floating around and sitting in the body. Another role is the reverse cholesterol transport, so plasma or tissue cholesterols are transported back to the liver. This reverse cholesterol transport mechanism reduces plasma cholesterols and also removes cholesterol from foam cells. So foam cells are a type of macrophage that localize to fatty deposits on blood vessel walls, where they ingest low-density lipoproteins and become laden with lipids, giving them a foamy appearance. And this is what we have right here. Get that plaque buildup, we'll have the foam cells and everything all in there. That's sort of the idea of what we're thinking about with atherosclerosis. Okay, functions and fate of HDL. So the newly made chylomicron only contains the lipids and apo48 as their lipoproteins, but does not contain apoC2 or apoE. Mature HDL, which contains apoC2 and apoE, serves as the repository of apoC2 and apoE for both chylomicron and VLDL, and donates the apoC2 and apoE to the newly formed chylomicrons and the newly formed VLDLs. As a result, the newly formed chylomicrons and new VLDL are converted to mature chylomicrons and mature VLDL, respectively, following receiving of apoC2 and apoE from the mature HDL. So this is showing all that, where the HDL is going to be giving those apoproteins to the specific ones. So giving the apoC and apoE to the newly made chylomicron that has potential to make it a mature chylomicron. The new VLDL also getting the apoE and apoC, apoC2 specifically, and so on, and then breaking down the HDL, picking up the other triglycerides and everything, and then either getting converted to an IDL or an LDL and coming back to the liver. Nascent HDLs containing only apoA are generated in the liver and intestinal cells. Mature HDLs are generated when they receive the recycled apoC2 and apoE from the processed chylomicron remnants and the processed LDLs. Mature HDL exchange apoC2 and apoE proteins with the newly formed chylomicrons, which contains only its lipid core and the ApoB48, and the newly formed VLDL, which only contains its lipid core and ApoB100, and make them mature chylomicron and mature VLDL containing that apoC2 and apoE on them. Thus, mature HDL serves as the donor of the apoC2 and apoE to chylomicron and the VLDL. Cholesterols embedded in the cellular membranes are picked up by HDL and is converted to cholesterol esters, or the CE, by the enzymatic action of the LCAT of HDL. Cholesterol esters are then transferred to the VLDL particles in exchange of triglycerides, and ApoD, or CETP, participates in the transfer of those cholesterol esters. So then the receptor-mediated endocytosis of lipoproteins containing the cholesterol, or the cholesterol esters, occurs. So clinical correlations are required. We're talking about hyperlipidemia. So hyperlipidemia type 1 is due to lipoprotein lipase deficiency or due to apoC2 deficiency and has high cholesterol levels. Hyperlipidemia type 2 is due to LDL receptor deficiency, causing high LDL, VLDL triglyceride levels. This is the one that we were talking about earlier with the what's called the the PCSK9 genetic, that gene. An issue with that gene will give us this type 2. Hyperlipidemia type 3 is due to defective ApoE. Hyperlipidemia type 4 is due to VLDL overproduction. Hyperlipidemia type 5 is due to VLDL overproduction and decreased LPL synthesis. We can also have LCAT deficiency, which causes an inability to form cholesterol esters from the HDL-associated cholesterol. This results in decreased ability to remove blood cholesterol and manifests as defects in the kidneys, red blood cells, and the cornea of the eyes. Okay, and that is our whole chapter 4, going over all of the different lipoproteins. So big things to watch out for, and remember kind of the structure of the overall lipoprotein, what they contain, the lipoprotein, the lipids themselves, the amphiphilic lipids, as well as then the apoproteins. You have the different types of apoproteins. You have our different types. Remember the densities, what the different densities are, what the role of each of these are, kind of where they're either created or where they're typically located, how when you the different functions of the apoproteins. What else is really important to remember? The function of each of the lipoproteins, those major roles. And yeah, that looks good. Okay, thank you guys for watching this video. Hopefully it helps you understand a little bit better about these lipoproteins. If you have any questions or comments, put them down below, and I will give to them as quickly as I can. And this actually should, if I'm not mistaken, let me double check, this should take us to what is included in exam 1, or right around here should be considered our exam 1 material. Yeah, according to my records, this is everything that's on exam 1. So I will look and see if I can find a study guide as well to go over and post a video going through a study guide of those major components, or we will review that ourselves as part of in-person tutoring. So yeah, have a great day.